Welcome to the deep dive. Our mission today, well, it's to look past modern borders, the lines on the map, and dive deep into history as it's written in our DNA. We're exploring the really complex, ancient genetic story of California, the U.S. state, and the Baja California Peninsula down in Mexico. We want to see how people north and south of that border have been connected for thousands of years.
Yeah, it's a truly remarkable area for studying ancient population dynamics. We've pulled together quite a range of sources here. We're looking at ancient paleogenomic data, including from really unique groups like the Paracues down in Baja California Sur, plus modern genetic studies of mestizo populations, and also detailed mtDNA work on California indigenous groups.
And what's the big picture emerging?
Well, what these sources show pretty clearly is that the story of human migration and adaptation and even isolation in this part of North America is, well, it's incredibly complex. It often defies the geography we just take for granted today. Oh, and just a quick note, when we say BP in our chat, that means before present, technically before 1950, just so everyone's on the same page.
Got it. Okay, so let's start maybe where people might expect a simpler story. Ancient California, north of the current border. The sources paint this picture of, wow, really deep continuity, almost like a family tree going back 7, maybe 8,000 years.
That's actually a perfect way to put it. The most striking thing really is the evidence for just incredible genetic stability along the Santa Barbara coast and out on the Channel Islands.
Yeah.
We're talking real genetic continuity here, backed up by genome-wide data. It stretches from about 7,400 BP right up, almost unbroken, to around 200 BP.
So essentially the direct ancestors of modern Chumash groups.
Exactly, and finding that kind of continuous deep time presence in one place. It's pretty rare in the global archaeological record. It's quite special.
And you can actually see that stability reflected in the maternal lines too, right? The mitochondrial DNA.
Precisely. When you look at the mtDNA from both ancient remains and modern Chumashan people, you see this clear dominance of specific haplogroups, mainly A and D. But the really compelling part, the proof of that long-term stability, is within their haplogroup A lines. It's not just one single line persisting. It's like a branching chain, just like that tree analogy you used. The lineages are diversifying over thousands of years, but all within that same relatively small area. It screams successful, stable population that wasn't just replaced by newcomers for a huge amount of time.
Okay, so there's this anchor of stability with the Chumash. But, California's history is never quite that straightforward, is it? The sources then bring in evidence of major movement coming from the south. And this is way before agriculture became widespread.
Yes. And this is where that ancient connection across today's borders starts to really take shape. Around 5,200 BP, you see this clear, noticeable uptick in genetic markers. These are distinctive lineages that are strongly associated with populations in northwest Mexico, and they start appearing in southern and central California.
5,200 years ago. That's ancient history. So what do researchers think might have been driving these people north? It wasn't farming yet, you said.
Well, the leading idea is that these groups are strong candidates for spreading Uto-Aztecan languages into the region, and that timing is absolutely crucial. This northward push happens before maize agriculture really spreads up for Mexico. So the drivers seem to be something else, maybe other social factors, cultural shifts, or changes in hunting and gathering strategies, not farming initially.
And we can still see the genetic echoes of those different major movements today.
Oh, absolutely. The distinct patterns are still there. You have the Chumash groups with their long history showing mostly haplogroups A and D. Then you have the groups linked to the Uto-Aztecan languages who predominantly show haplogroups B and C. It's like a genetic fingerprint differentiating those two major deep historical layers. Stability, and then this significant influx from the south.
All right, let's pivot now and head south, down the Baja California Peninsula. Geographically, it kind of acts like a long cul-de-sac, right? We're focusing on the southern tip, Baja California Sur in Mexico, home to the ancient Paraque people. The sources describe them as, well, maybe one of the most genetically isolated groups ever studied from that pre-Hispanic period.
Isolated almost doesn't capture it. Yeah, they were hunter-gatherers, and their genetics basically scream closed population for a very, very long time.
Okay, isolation and genetics. Sometimes the technical terms fly over my head a bit.
Okay.
Can you break down what that looks like conceptually before we hit the metrics?
Think of it like this. Imagine the Paracues living on a really remote island. Even though they were on a peninsula, it was hard to get there, hard to leave. Because their group size was small and they rarely, if ever, mixed with outsiders for probably long stretches of time, the genetic diversity within the group just shrinks. And certain stretches of DNA become identical between individuals. Basically, they became highly inbred over generations, not by choice, but because there just weren't other options.
Right, so what's the hard evidence? The technical proof for that level of profound isolation?
Researchers use a couple of key genetic measures. First, something called conditional nucleotide diversity. The Paracues had the lowest values by far compared to other ancient Mexican groups. That confirms the lack of overall genetic variation, low diversity.
Okay.
Second, and maybe even more revealing, is an analysis of runs of homozygosity ROH for short. Think of your chromosomes coming in pairs, like two copies of a book. In a small, isolated population, those books start having long, identical passages where the text matches perfectly page after page.
Right.
The sources found the Paracues had the highest total length of these long, identical stretches, ROH greater than 4 centimorgans, which is just a unit of genetic distance. Really high ROH points directly to high levels of relatedness, inbreeding, stemming from a small effective population size over time.
So the high ROH is like the genomic signature confirming that remote island idea you mentioned.
Exactly. Their ROH levels were actually quite similar to what's been found in ancient hunter-gatherers from Patagonia, way down in South America, another group known for extreme isolation. Though, interestingly, the data hints the Paracu population might have been slightly larger overall than the Patagonian group studied, but still they're a major genetic outlier, a snapshot of stability achieved through deep isolation.
Before we connect the Paracues back north to California, maybe just a quick word on the modern indigenous landscape in Baja. It's not an empty place.
Oh, absolutely not. It's important to remember that today Baja California is home to several indigenous groups, mainly belonging to the human language family like the Kukapa, Killua, Pai, and Kumeyaay, and also the Cochimi people. It's actually a really positive note that there's active cultural revitalization happening, including among the Cochimi, who were sadly thought to be culturally extinct back in the 19th century. Their resilience is part of this long, layered story, too.
Okay, now for the part that really ties this all together, the historical twist. We've got the stable Chumash lineage up in California. We've got the Uto-Aztecan-related groups arriving around 5200 BP, and then these incredibly isolated Paracues way down south in Baja Sur. What happens when you compare the Paracu DNA with those early Californians?
This is where the deep dive really turns things on our head. It's quite startling. The ancient Paracu individuals from Baja Sur share more genetic drift, basically, more rare alleles, more shared ancestry with the oldest Central Californian individual they analyzed.
The one from 5200 BP.
Yes, the one from near Pacific Grove, dated to about 5200 BP. They share more with that single ancient individual than they do with any of the later Central Californians, people who live in the same area just a few thousand years after them.
Hold on, let me make sure I heard that right. You're saying an ancient person in Central California is genetically closer to this super isolated group hundreds and hundreds of miles south across deserts and water. Closer to them than to the people who lived in his own region later on. That sounds backwards, given how isolated the Paracues were supposed to be.
It is genuinely shocking and counterintuitive at first glance. But the data is quite clear. It strongly suggests that this Paracu-related ancestry, whatever it represents, was actually quite prominent in the very early populations settling coastal California. But then over time, that signal got diluted, almost washed out. It's like an old radio station that was broadcasting strongly at the beginning, but then got drowned out by newer, more powerful stations transmitting from different directions, maybe from inland North America, perhaps. So this early genetic thread connecting Baja Sur and early Central California, it might be a trace of an even earlier founding population, or maybe what some linguists call a substrate, an older language layer whose genetic signal faded as later groups, like those potential Uto-Aztecan speakers, moved in and became dominant.
That's precisely the implication researchers are drawing. It points towards this ancient, almost vanished genetic bridge. It suggests the earliest peoples in that part of California might have been closely related to the ancestors of the group that eventually became trapped, genetically speaking, at the southern tip of Baja. And then later migrations effectively overwrote much of that initial genetic signature.
Wow. That really underscores just how dynamic and layered this ancient history must have been. Okay, let's shift now towards the modern day, thinking about how all these deep histories, plus the impact of European colonization, shape the genetics we see today in Mexico, particularly in the admixed mestizo populations, which are often studied for things like genomic medicine.
Right, so when researchers look at the average genetic makeup of modern Mexican mestizos, the breakdown is pretty consistent across large studies. It averages out to about 55.2% indigenous Amerindian ancestry, 41.8% European, and then smaller bits of African, around 1.8%, and East Asian, about 1.2%. That average, of course, reflects centuries of mixing across a huge and diverse country.
But the sources point out a really stark contrast when you look specifically at the maternal line, the mtDNA, which only comes down from the mother's side.
Yes, and this is absolutely crucial for understanding the human side of colonization. Even though the overall average ancestry is nearly 42% European, if you look only at the mitochondrial DNA of modern Mexicans, almost 90% of it belongs to one of the four major indigenous American haplogroups, A2, B2, C1, or D1.
90%. That's a huge discrepancy from the overall 42% European number. What does that tell us?
It paints a very clear demographic picture. The primary pattern of European colonization, genetically speaking, involved European men mixing with indigenous women. There was a very, very low contribution from European women to the modern Mexican gene pool, at least on the direct maternal line. It means indigenous maternal lineages were incredibly resilient, genetically speaking, through the entire colonial period and beyond, preserving that heritage on the female side on a massive scale.
And just to add another layer of ancient complexity, the research also hints at populations we haven't even found physical evidence for yet. These ghost populations.
Yeah, this is fascinating cutting-edge stuff. It's speculative in the sense that we haven't found their bones, but it's based on solid genomic analysis. When scientists sequence the DNA of pre-Hispanic individuals, say from the Sierra Tarahumara in northern Mexico, or a site called Cañada de la Virgen in central Mexico, they find bits of ancestry in their genomes that just don't match any other known ancient or modern group that has been sampled.
So there's like an unknown ingredient in their genetic recipe.
Yeah. Exactly. An unsampled population contributing to their makeup. They've even given it a placeholder name, UPOB-A, for unsampled population A, for the one found in northern Mexico. But here's the really interesting part. The ghost signal found in central Mexico seems to be different. They call it UPO-A2. This suggests there wasn't just one unknown ancient group, but potentially multiple distinct populations contributing to Mexico's deep history that we just haven't identified yet.
That really opens up the possibility that our map of ancient American genetics is far from complete. Now, doing this kind of research, working with ancient human remains, it comes with a history and ethical considerations that need talking about, right?
Absolutely, and the sources do bring this up. It's important context. Feels like American anthropology and archaeology, well, they grew up alongside colonialism.
Yeah.
And frankly, in the past, some research was used to diminish or misrepresent Native American history and diversity.
Right.
And for researchers today, particularly in places like Mexico, where the national story often emphasizes mestizaje, this idea of a single mixed national identity, it adds complexity. How do you ethically study ancient remains that specific indigenous communities today view as their direct ancestors, especially when the broader narrative sometimes downplays that unique ancestral connection? It requires careful navigation and consultation.
And dealing with the legacy of destructive analysis.
Yeah.
You know, destroying a piece of bone or tooth to get the DNA, often without prior proper consultation with descendant communities, that's a big focus now, isn't it?
It's essential. There's a strong push across the field towards more collaborative, equitable partnerships. Recognizing the harm done when science proceeded without community consent is step one. It's really about ensuring that the communities whose ancestors are being studied have a real voice in that process, respecting their connection, their history, their concerns.
Well, that brings us towards the end of this deep dive. We've journeyed from the really striking long-term stability of the Chumash in California, reinforced over millennia, down to the incredible isolation of the Paracues in Baja Sur, and then uncovered this faint, ancient genetic thread connecting them, a thread largely obscured by later migrations from inland areas.
Yeah, the big takeaway for me is just how much more intricate and dynamic the peopling life of North America was than our simple maps might suggest. You have this complex interplay of stability here, migration there, subsequent ways diluting earlier signals, and then the massive impact of colonization layered on top. It really challenges us to keep revising the story. And considering that ghost ancestry we talked about, those unsampled UPOP and UPOP lineages hinting at hidden populations in ancient Mexico, well, it leaves us with a pretty powerful final thought for you, the listener, to chew on.
If we've only scratched the surface of sampling the genetic diversity of ancient peoples across the Americas, what other completely unknown populations, what other major migrations, what other ghosts are still out there, waiting in the soil or the data, ready to completely rewrite our understanding of the continent's deep human past?